All right, welcome back. Today our topic is going to be all about enzymes and their role in living things, as well as the type of reactions that they assist with. So enzymes help with chemical reactions, and all of life is built on chemical reactions. Um, and enzymes are going to assist with these chemical reactions, and they're necessary for helping maintain metabolism and homeostasis. So if we flash back to Unit 1, we talked about the characteristics essential for life. And two of those characteristics were metabolism and homeostasis. Now remember, metabolism was the ability to regulate energy. And there are different types of metabolisms that an organism could have. Organisms could be producers or autotrophs, meaning that they make their own food. And then that food will be used to make energy. Or an organism could be a heterotroph or a consumer, meaning that they rely on other things to get their food. So that was metabolism. And homeostasis was the ability to maintain a stable, stable, internal condition. So that would be like maintaining your temperature, maintaining your blood pressure, and your heart rate. And both of these, metabolism and homeostasis, are essential for life. So metabolism, the regulation of energy, is controlled by all of the chemical reactions happening in your body. So metabolism, all the chemical reactions in your body that help regulate and use and process energy. And enzymes are going to help with these reactions. And reactions typically can do one of two things. They can help to make or form a bond. So forming bonds between two molecules. An example of that would be dehydration synthesis. We talked about that a little earlier in this unit. Dehydration synthesis takes away water to make a bond. So this would be essential for building a polymer or a macromolecule. So if we did not have enzymes, we wouldn't be able to build our macromolecules. And they are also essential for breaking down molecules. And we talked about a type of reaction called hydrolysis that breaks down molecules. And this happens by adding water to break a bond. And the reason why we would need to break bonds would be in the event that we eat food. So we eat food, we need to be able to break it down to be able to use it. And enzymes are going to help break down molecules so that we can use the nutrients stored in that molecule. And this is going to come down to two different types of reactions. Some reactions are going to release energy, and they're called energy releasing. And typically energy releasing reactions are going to be ones that break bonds. So ones that break down polymers or break bonds, such as the hydrolysis reaction, are going to be ones that release energy. And this is because energy is stored in bonds. So when you break a bond, you're releasing the energy stored there. So if we look at this graph here, graph number 1, this would be an energy releasing reaction. We had put some in, but in the overall end, energy is released. A second type of reaction would be an energy absorbing reaction. These are going to be reactions that require an input of energy to perform. Typically, reactions that involve putting in energy are going to be ones that build polymers or make bonds. Making bonds requires putting in energy. And the example that we talked about would be the dehydration synthesis reaction that makes bonds. And those require energy. Graph 2 shows an energy absorbing reaction, because in the end you have to put energy in for it to be complete, and you never make it down to the energy released stage. Now you may ask, if reactions are downhill, why don't they just happen spontaneously? So why would it need help of an enzyme? Why wouldn't it just happen randomly? And that's because if we are releasing energy in the end, we are breaking bonds. And we talked about earlier this unit, when we talked about bonds, we talked about covalent bonds being stable. So covalent bonds are stable bonds, meaning it's going to take energy to break the bond. So while this reaction here in graph 1 does release energy in the end, it requires an input of energy to get it started. So it can't just happen randomly. It does require an input of energy. But once the bond itself is broken, it releases enough energy to compensate for the input. So while it releases energy overall, it does require energy to be put in to break it first. So that's why you're not just happening spontaneously, because it requires that initial input of energy in order to break the bond to get any energy out in the end. Now, the energy that you have to put in the beginning is called the activation energy, or the energy needed to start the reaction. On this graph, the activation energy is the distance from this line where you start to the very top of the graph. I have to overcome this peak here in order for my reaction to proceed downhill. So essentially you can think of this as like a speed bump or an energy hill. I like speed bump better, because once you get over it, everything is downhill, but you do have to put in that initial energy to get over the hump in order to get the energy out in the end. So this brings us back to enzymes. Now we're going to talk about enzymes specifically. We talked about types of reactions. We said enzymes help out, but we've never said what they are or what they actually do specifically. So enzymes are special proteins. Now, back in our last session, we talked about the macromolecules. Proteins were a macromolecule made of amino acids, proamino, so they are made of amino acids. A protein is a chain of amino acids linked together. Once the amino acids are linked together, they will then fold to form a protein that has a specific shape. We said that enzymes are highly specific, so that is right here. And they can only help with one type of reaction. So enzymes facilitate means to help. They help chemical reactions by increasing the rate, so they make it go faster. So increase rate of reaction, that means make it go faster. So they help the reaction happen faster. Now, enzymes are highly specific, meaning one enzyme can only help one job happen. One enzyme can't do many different things. It can only do one specific type of thing. And enzymes are required for most reactions in the body. So most reactions in the body have a specific enzyme that helps out. So this brings us to another term called a catalyst. A catalyst in general, all alone, is a chemical agent that helps chemical reactions by reducing the amount of activation energy. So it's actually missing right here. So we're going to change that to lowering or reducing. So a catalyst is any chemical agent that helps a reaction by lowering the activation energy. So what this means is that a catalyst is going to lower the speed bump. And this is what an enzyme does. An enzyme is a protein that lowers the activation energy of a reaction. That means that enzymes are biological catalysts. So if I was to go back up here a little bit, here would be the normal reaction without an enzyme. The speed bump here is very high. See how high the speed bump is here? The activation energy is fairly high. What an enzyme does, so here's a graph of a reaction with an enzyme, and it takes my activation energy and lowers it significantly. Look how much my speed bump lowered. It used to be up here, now it's down here. So the enzyme lowered the speed bump or lowered the activation energy for the reaction to happen. That means you have to put in less energy, so therefore the reaction is going to happen much faster. So enzymes lower activation energy, they lower the speed bump, and lowering the activation energy speeds up the reaction. And the reason this happens is, let's say you're going over a speed bump in your car. So if this is you in your car, and you can either go over speed bump A or speed bump B, and you want to go fast, the one that you're going to be able to go faster over is going to be speed bump A. You went faster over A than you did over B. You could also think if this is you and you are about to climb a hill, the hill you're going to want to climb and the hill that you can climb the fastest is hill A. You're going to be able to climb A a lot faster than you can climb hill B. So we're going to think of activation energy like a speed bump or a hill. The lower the bump or the lower the hill, the faster you can go over it. We're going to work on Canvas. We're going to work on labeling these graphs and understanding them a bit further. So some specific vocabulary behind enzymes and how they work. The first word that you'll need to know is substrate. A substrate is the reactant that the enzyme bonds to. So if I have an enzyme and it wants to break a bond, I'm going to have the substrate, which the enzyme is going to help break in half. So substrate would be like the reactant, the thing that goes in that needs help. So here is my substrate here in dark blue. Light blue is my enzyme. The way the enzyme is going to help is they're going to bind together. The substrate and the enzyme are going to sit perfectly together. When they are connected, when they are together, they're called the enzyme substrate complex. This is when the enzyme is actually doing its job. So the enzyme substrate complex is formed when the enzyme and the substrate bond together and they're stuck together. The special shape, if you notice here, the enzyme has a special shape that perfectly fits the substrate. This is going to be known as the active site. It's the site on the enzyme that does a job. It's the active site. It's going to have a specific shape that fits the substrate perfectly. So again, your substrate and your enzyme come together. The reaction happens. Our goal was to break a bond. Once the bond is broken, once the enzyme does its job, then we're going to be left with products, which are going to be the end results from the reaction. And again, this is what it looks like when it comes in. You have your substrate, which is the molecule that the enzyme needs to help. You have your enzyme. The enzyme has a very specific shape called the active site. The substrate is going to fit right into the enzyme's active site. They're going to bond together. When they're together, it's called the enzyme substrate complex. The enzyme does its job, breaks the bond, and the products are released. Okay, that was a lot, so we're going to go over just a quick review here. What is activation energy? The activation energy is the energy needed to start the reaction. It's like the speed bump you have to overcome. How do enzymes actually lower the activation energy and then speed up the reaction? We said there were different types of reactions that enzymes could help out with, and they could either help create a bond or they could help break a bond. So enzymes just come in and they help. They're helpers. They help reactions happen. We said that enzymes are very, very specific. They have a specific shape at the active site so that only one type of substrate can fit in. Something new? Enzymes are actually named for the reactions that they help catalyze. Remember, catalyze means, so enzymes are named for the reaction that they help happen. And their name usually ends in ASE. So for instance, this one, sucrase, it breaks down sucrose. Lipase breaks down lipids. DNA polymerase builds DNA. So they are often named for the reaction that they help happen. Try to keep it as specific as possible. So we had this complex image here about exactly how enzymes help out. The easy way to think of it is like a lock and a key. So everybody who has a car has a key for their car. So everybody that has a car key, everybody has a key. Keys look similar, but not all keys fit the same car. Like my car key isn't going to start your car, or my house key isn't going to get into your house. Each one is very, very specific, and they have to fit together perfectly in order to work. The same is true for an enzyme and its substrate. So an enzyme has one shape at the active site. The substrate has to fit it perfectly. So the enzyme would be the lock, and the substrate would be the key that's going to fit into the lock. One thing to keep in the back of your mind, enzymes are not consumed in a reaction, meaning they're not used up, they don't disappear. Enzymes, they're very specific, meaning they can only help one type of reaction, but they can do that one reaction thousands of times. And that's just per second. So, for instance, if we were to go back up here to this list, let's look at these two. Sucrase breaks down sucrose. Lipase breaks down lipids. Sucrase, the enzyme, can break down thousands of molecules of sucrose a second. But it will never, ever, ever be able to break down lipids, because it can only do one type of job. So your key will unlock your door thousands of times, but someone else's key is not going to unlock your door ever. Okay, so we talked about this a little bit with proteins. We're going to go over it one more time. We talked about factors that affect how enzymes work. So we discussed how temperature and pH can both affect how an enzyme works. Each enzyme is going to have its own unique environment where it works best. So different enzymes are going to function best in different environments. So enzymes in my stomach are going to work differently than enzymes in my mouth. They're going to be very different. The word optimal means best. So each enzyme has its own optimal conditions, meaning conditions in which it works best. So if we were to look at here, this graph that we have here, we have rate of reaction on the Y and pH on the X. This enzyme will work best at a pH of 2. This enzyme in red is going to work best at a pH of 8. So every enzyme will work best in different conditions. Each enzyme has its own specific environment that it's going to do best in. What that means, if I take an enzyme out of its natural environment and put it somewhere else, it's not going to work as well. So if you change the condition of an enzyme too much, it will change its shape, and it will become denatured. If an enzyme denatures or changes its shape, it can no longer work. So if we look at the image over here on the PowerPoint, if the enzyme is supposed to have this shape, but then it changes its shape because you change the temperature or pH, now the substrate can't fit, and if the substrate can't fit, the enzyme doesn't work. So if you change the temperature or if you change the pH too much, the enzyme will change shape and will not be able to do its job. In our last video, I equated this with eggs. So again, if you break an egg, a raw egg is going to have the yellow yolk, and it's going to have the clearish jelly-like substance as well. If you were to heat an egg in a pan, it looks different because it changes, the enzymes change shape. The same thing happens if you put an acid or a strong base on an egg. It changes its shape and it becomes completely white instead of having that clearish, gooey color. And again, you can't uncook an egg, meaning once the enzyme changes its shape, once it denatures, you can't fix it. Okay. So quick review, what is a catalyst? A catalyst is a substance that is going to help speed up a reaction. Activation energy. The activation energy is the energy needed to start a reaction. How do enzymes speed up chemical reactions? They speed up chemical reactions by lowering activation energy. So enzymes speed up chemical reactions by lowering activation energy. And then the last question, what two factors can affect an enzyme's function? Those two factors are temperature and pH. Okay, so make sure you answer these on your sheet. If you need to pause and go back and listen again, feel free to do so. There's going to be some practice after this on Canvas.